Hello class, today we are going to discuss about blood grouping and structure of heart. Blood grouping mainly ABO blood grouping and Aris blood grouping. The person in this picture is Carl Landsteiner. He discovered ABO blood group system in 1901. He also discovered Aris blood group system along with Alexander Wiener in 1930 and he was awarded the Nobel Prize in 1930. So let's study about ABO blood group. In ABO blood group system, there are four blood groups A, B, AB and O. And this system is based on the presence or absence of antigen A and B on the surface of RBCs. Let's make it simpler. Here you can see a person of A blood group has antigen A on the surface of RBCs. A person of B blood group has antigen B on the surface of RBCs. A person of AB blood group has both the antigen A and B on the surface of RBCs. And a person of O blood group has neither antigen A nor B on the surface of RBCs. As we all know, the plasma of different individuals have two natural antibodies, anti-A and anti-B. See, in case of A blood group, the antigen is A, but the antibody in plasma is anti-B. In case of B blood group, the antigen is B, but the antibody in plasma is anti-A. In case of AB blood group, where both the antigens are present, both the antibodies are absent in plasma. In case of O blood group, where both the antigens are absent on the surface of RBCs, but here both the antibodies anti-A and anti-B are present in plasma. So this A blood group can receive blood from A and O blood group. B blood group can receive blood from B and O blood group. The AB blood group can receive blood from all the four groups A, B, AB and O. And the O blood group can receive blood only from O group but can donate blood to all the four groups. Therefore, this AB blood group is known as the universal recipient, while the O blood group is known as universal donor. So, now the question is, in blood transfusion, why it is so important to match the blood group of the donor and the recipient? The answer is, in case of incompatible transfusion, what will happen? The recipient makes antibodies against the antigen which are present on the surface of RBCs, and these antibodies attack the antigen, as a result of which hemolysis occurs, destruction of RBC occurs, which may result in death. See, here you can see, if a blood group A receives blood from A group, here in case of blood group A, the antigen A is present on the surface of RBC, while anti-B antibody is present in plasma of the recipient. This anti-B antibody cannot combine with the antigen A of the donor. Therefore, there will be no clumping, no destruction of RBC. So this type of blood transfusion is safe. But in this case, if a B blood group receives A type of blood, what will happen? In case of A blood group, antigen A is present, and in case of B blood group, the anti-A antibody is present in plasma. So these anti-A antibodies bind with the antigens which are present on the surface of RBCs, causing the clumping of blood or destruction of RBCs. So it is very important to match the blood group of the donor and the recipient before any blood transfusion. So let's discuss about Aris group. This blood group system was also discovered by Carl Landsteiner along with Alexander Wiener in 1930, and this blood group is based on the presence or absence of another antigen Rh on the surface of RBCs. See, if the Aris antigen is present, the blood group is Aris positive. If the Aris antigen is absent, the blood group is Aris negative. See, in case of O positive blood group, only the Aris antigen is present. In case of A positive, both the antigen A and Rh are present on the surface of RBCs. In case of B positive blood, the Rh antigen along with the B antigen are present on the surface of RBCs. In case of AB positive, all these three antigens A, B and Rh are present on the surface of RBCs. And in case of O negative blood, all these three antigens are absent. In case of A negative blood, only antigen A is present. In case of B negative, only the B antigen is present. In case of AB negative, A and B antigens are present. So, Aris positive blood can be donated only to Aris positive blood type, but it can receive blood from both the Aris positive and Aris negative blood type. And the Aris negative blood can be donated to both the Aris positive and Aris negative blood type, but it can receive blood only from the Aris negative blood type. See, the AB positive blood group, can receive blood from all the 8 blood groups, that is A positive, A negative, B positive, B negative, AB positive, AB negative, O positive and O negative. But this type of blood can be donated only to the AB positive blood type. Therefore, if we can say precisely that this AB positive blood group is universal recipient. Okay. Now this O negative blood, you can see this O negative blood group can receive blood only from the O negative type, but it can donate blood to all the 8 blood groups, the A positive, A negative, B positive, B negative, AB positive, AB negative, O positive and O negative. Therefore, this O negative blood group is known as the universal donor, and this is a very rare blood phenotype. Only 7% of the population have this O negative blood type. You remember, this is the safest blood for any transfusion. So, let's discuss a condition which is known as Aris incompatibility or Aris mismatching. This condition occurs where a Aris negative mother is expecting a child of Aris positive blood. Let's understand it simply that when the father is Aris positive and mother is Aris negative and the fetus is Aris positive, during the first pregnancy, what happens? This placenta ensures no mixing of the fetal and maternal blood. But during childbirth, during parturition or childbirth, a little amount of fetal blood mixes with the maternal blood. So now this maternal blood receives the Aris antigen, and obviously in response to that Aris antigen, the mother's body now will produce anti-Aris antibodies. And these anti-Aris antibodies are of immunoglobulin Z type. We all know there are 5 different types of antibodies: immunoglobulin Z, immunoglobulin M, A, B and E. And these anti-Aris antibodies are of immunoglobulin Z type. See, what will happen during the second pregnancy? The fetus is again Aris positive, and these anti-Aris antibodies are already in mother's maternal blood. So these anti-Aris antibodies are of immunoglobulin Z type. We know this is the only class of antibody that can cross the placenta. So during the second pregnancy, these antibodies will cross the placenta and enters into the fetal blood, and the anti-Aris antibodies will attack the Aris antigen of the fetal blood, as a result of which hemolysis will occur, destruction of RBCs will occur, causing severe anemia and jaundice to the newborn. See, during first pregnancy it is safe, but during second pregnancy it will cause anemia and jaundice to the newborn, and this condition is known as erythroblastosis fetalis or hemolytic disease of the newborn. So how this condition is treated? To treat this condition, to prevent the Aris immunization, the Aris negative mother is given an injection, intramuscular injection, this one, Rhogam. Okay, Rhogam is an intramuscular injection which is given to the Aris negative mother at 26 to 28 weeks of pregnancy and after the delivery of an Aris positive baby. So how Rhogam works? This Rhogam prevents the Aris negative mother from making antibodies against the Aris antigen. Therefore, if the maternal blood, if there is no anti-Aris antibodies in the maternal blood, there will be less possibility to cause hemolytic disease to the newborn. So as long as the Aris negative mother receives the Rhogam appropriately during her pregnancy, her babies are at lower risk of developing the hemolytic disease. Students, we have discussed about the ABO grouping, Aris mismatching condition. Now we are going to discuss about a special blood group which is called the Bombay blood group. And this blood group was discovered by Dr. Y.M. Phinde in 1952. And this type of blood group is deficient in expressing the H antigen. So what is H antigen? We have already discussed about A antigen, B antigen. So what is H antigen? This H antigen is the precursor of all the ABO blood group antigens. If this H antigen is not produced, all ABO blood group antigens are also not produced. Whether a blood group is AB or O, H antigen is present in all the blood groups. But in this blood group or in this blood type, it lacks antigen A, B and H, but the plasma contains anti-A, anti-B and anti-H antibodies. And this type of blood group can receive blood only from the same blood type, but it can donate blood to all the blood types. Sometimes the blood group is confused with the O blood group, but in case of O blood group, the H antigen is present, but in case of Bombay blood group, the H antigen is absent. So students, now we are going to discuss about the structure of heart. We know the human circulatory system is consist of a muscular chambered heart, a network of close branching blood vessel and blood. So we are going to discuss about the structure of heart. We know our heart is mesodermal in origin, that is, it is a mesodermally derived organ, and it is located in the thoracic cavity in between the two lungs, and the space in between the two lungs is known as mediastinum. And remember, our heart is tilted to the left, slightly tilted to the left. So our heart has a size of a clenched fist, clenched like this. So the heart is protected by a double membranous bag, which is known as pericardium. The outer pericardium, which is known as parietal pericardium, and the inner pericardium, which is known as visceral pericardium. And this pericardium, this membrane encloses the pericardial fluid. So what is the function of pericardial fluid? This fluid reduces the friction during the contraction and relaxation of heart. So contraction and relaxation of heart. So we know our heart has 4 chambers, 2 upper chambers which are relatively smaller and 2 lower chambers. The upper chambers are called the atria, the right atrium and left atrium. The lower chambers are called the ventricles, the right ventricles and left ventricles. Both the atria are separated by a thin muscular wall which is known as the interatrial septa, and both the ventricles are separated by a thick muscular wall which are known as interventricular septum. So again, the atria and ventricles of the same side also separated by thick fibrous wall which is known as the atrioventricular septa. And these septa, each of these septa are provided with opening so that the chambers are connected, and the opening in between the right atrium and right ventricle is guarded by a valve, the tricuspid valve. And the opening of the left atrium and the left ventricle is guarded by another valve which is called bicuspid valve. You just remember 2 words, right and left. Right means right atrium tricuspid, left means left atrium bicuspid. This bicuspid valve is also known as the mitral valve. So again, the opening of the left ventricle to aorta is guarded by semilunar valve, and the opening of the right ventricle to pulmonary artery is guarded by another semilunar valve. So what these valves do? This valve maintains the blood flow in one direction, that is from atrium to ventricle and from ventricle to aorta and pulmonary artery. It prevents the backward flow of the blood. Students, we have discussed about the chambers of heart. Now we know that our heart is made up of a special muscle, the cardiac muscle, and a specialized musculature is also distributed in our heart, which is known as nodal tissue. Here you can see a patch of nodal tissue is situated A patch of nodal tissue is situated on the right upper corner of the right atrium, which is known as the sinoatrial node or SA node. This SA node can generate the maximum number of action potential, which is 70 to 75 per minute. And this SA node is responsible for initiating and maintaining the rhythmic contractile activity of our heart. Therefore, this SA node is also known as the pacemaker of our heart. So another patch of nodal tissue, another patch of nodal tissue which is located on the lower left corner of right atrium. This is called the atrioventricular node or AV node. A bundle of fibers continues from AV node, here you can see, and emerges on the top of the interventricular septum, and then it divides into two bundles, and these fibers give rise to Purkinje fibers. And these Purkinje fibers or these nodal fibers along with the right and left bundle is known as bundle of His. So I think we have covered almost all important points about the structure of heart. So let's see the diagram. This is the aorta. The aorta carries oxygenated blood from left ventricle to all the body parts. This is pulmonary artery. The pulmonary artery carries deoxygenated blood from right ventricle to lung. These are pulmonary veins. Pulmonary veins carry oxygenated blood from lung to left atrium. This is left atrium, then bundle of His. We have already discussed about this. Then left ventricle, interventricular septum, and tip of heart. This structure, tip of our heart, is known as apex. Here you can see the right ventricle. Okay, this is chordae tendineae. You just remember chordae means cord and tendineae tendons. These are some strong fibrous connection in between. Here are the papillary muscle, in between the papillary muscle and the flaps of the valves. Then you can see the atrioventricular node. This one is AV node, and this right atrium, then SA node, and vena cava. Vena cava, the superior vena cava and inferior vena cava. Remember, the inferior vena cava is guarded by a valve which is known as the Eustachian valve, and this vena cava, superior and inferior vena cava, carry deoxygenated blood from all our body parts to the right atrium. So students, I hope you all have a decent understanding about blood grouping, ABO blood grouping, Aris grouping, then Aris incompatibility and structure of heart. So with this I am concluding today's class. Thank you.